Hello everyone, my name is Gary O'Mealy, and in this video we are going to talk about target cells versus non-target cells. So you'll recall that we've been talking a lot about cell-to-cell communication thus far, and you'll also hopefully remember that the ability of two cells to communicate with each other in the body, regardless of how much distance separates them, is going to depend on the relationship between, number one, the ligand that is secreted from the signaling cell. And remember, this can either be a hormone, it can be a neurotransmitter, or it can be something else. But it's going to be usually a small molecule that travels towards the target cell, and what makes the target cell a target cell is that it possesses the correct protein receptor that binds to that particular ligand. So this applies to all types of cell signaling, but in the coming videos we're going to really start focusing on endocrine signaling, so what I'm about to say specifically relates to that. But because endocrine signaling specifically involves depositing your hormone into the bloodstream, and the bloodstream is going to distribute this hormone far and wide in the body, and most cells in the body are going to have access to what is in the bloodstream, not just nutrients and oxygen, but also hormonal regulators that are in the blood. That would indicate to you that the risk for non-specific effects of the hormone is very high. For example, we understand that insulin is supposed to affect very specific parts of the body like adipose tissue, skeletal muscle, and the liver. But if we're secreting insulin into the bloodstream, that insulin is going to get distributed to the blood that's around the brain, the blood that's around the heart. So the question becomes, how do we ensure that the hormone that gets deposited into the blood only affects its intended targets? So in this illustration, we look at three different cells that exist in a tissue, and these three cells have access to the blood supply, which you can see is down here. Now, what you're going to notice is that each of these three cells has a different repertoire of protein receptors. And what this means is that these three cells will respond differently to hormonal signals. So in this instance, we have one particular hormone that is in the blood, which is indicated by the red circles. You can clearly see what is supposed to be the correct receptor for this particular hormone. And true to form, only a cell with that correct receptor will be able to respond to this particular hormone. So if you look here, these hormones that are in the bloodstream exit the bloodstream, go into the interstitial fluid, diffuse towards their target cells, and then bind to their intended receptor. You'll notice that only cells 1 and 3 have the correct receptor for this particular hormone, and as such, they will be the only cells that will be able to produce a response to that particular hormone. Cell number 2 here, you will notice, has a receptor, but it is not the correct receptor for this particular hormone. That makes it a non-target cell and means it will not be able to respond to this hormone, even though that hormone is in very close proximity to this non-target cell. Okay, let's look at a different scenario. Now we have a completely different hormone that is moving through the bloodstream. Again, you can clearly see which receptor appears to be the correct match for this particular hormone. So when this hormone diffuses out of the bloodstream, it binds to its target, which is cell number 2. Cell number 2 is the target here, so it is going to produce the response. And since cells 1 and 3 do not have the correct receptor for this particular hormone, that makes them non-target cells and they will not be able to respond to this particular hormone, even though they were able to respond to the other hormone that we were looking at before. Keep in mind, there's all different types of hormones, there's all different types of neurotransmitters and other chemical signals. What makes a cell capable of responding to a particular signal is having the right receptor for that particular signal. So just because cells 1 and 3 were the targets for the red hormone, that doesn't mean they're going to be targets for the purple hormone. And similarly, just because cell number 2 was not a target for the red hormone, that doesn't mean it's not going to be a target for the purple hormone. Okay, and we're going to touch on this in a future video, but specifically when you're talking about hormones, there's going to be more than one hormone in the blood at any given time. There's actually probably going to be dozens of hormones in the blood at any given time. So in this example, we have both a red hormone and a green hormone in the bloodstream at the same time, meaning that these hormones will together diffuse towards their targets in the interstitial fluid. Cell number 1 only has a receptor for the red hormone, meaning that it will produce what we'll call response A. Response A is a response that is dictated specifically by hormone, the red hormone. However, you'll notice that cell number 3 has receptors for both the red hormone and the green hormone. This can mean one of two different things. First, it could mean that the cell is simultaneously going to produce two different responses. Response A, which is the exact same as what cell number 1 did, and response B, which is something that is unique to a cell that possesses the receptor for the green hormone. But what is also possible, and it's something that we'll talk about when we start talking about hormonal interactions in the future, is that there may be some synergy between the red hormone and the green hormone, meaning that you may get a stronger response with both the red and the green hormone together than you would with either hormone separately. So what is being presented here with the red and the green hormone presenting two different responses, that may be kind of a simplistic way of looking at it. So just kind of keep that in mind for the future. And then, of course, cell number 2 does not have a receptor for either the red or the green hormone, and that makes it a non-target cell for both hormones, and it will not produce a response. Okay, so obviously we used an example of hormonal regulation to illustrate the difference between a target cell and a non-target cell. But keep in mind, this concept applies to any situation in which a cell signals to another cell with a ligand. It's just an issue that is particularly important for hormonal regulation. But just to make sure you get the idea, let's look at a neural example. So the nervous system has effectors all over the body, muscles, glands, and other tissues alike. But some of those neural effectors may be innervated, meaning that a neuron synapses with them, but some effectors may be innervated by more than one kind of neuron at a time. And because of that, the tissue or the effector may be subject to multiple neurotransmitters at different times. So the best example of this is the autonomic nervous system. We're going to learn all about that in a future video. But what we're looking at here is a tissue effector. This is the SA node of the heart, or the sinoatrial node of the heart. The sinoatrial node of the heart consists of pacemaker cells that help to determine what a person's heart rate is. Now, the way in which we raise our heart rate and lower our heart rate according to systemic demands and conditions throughout the body is through the actions of the sympathetic division of the autonomic nervous system. So you can see an axon of a sympathetic motor neuron right here, which is synapsing with the SA node. And then the parasympathetic division of the autonomic nervous system, for which you can see the axon of a parasympathetic motor neuron here. So these cells of the SA node are synapsed on by two different neurons at the same time, one from the sympathetic division and one from the parasympathetic division. So the idea here is that the sympathetic motor neuron and the parasympathetic motor neuron are each going to release different neurotransmitters in order to differentially regulate a person's heart rate. So let's look at how that works. Now, the first thing I want you to notice is that each cell here possesses both red receptors and green receptors. Now, the way I illustrated it here is going to make you think that the cells on the right side have the green receptors and the cells on the left side have the red receptors, but that's not the case. I was just a little limited artistically here. Go ahead and assume that every single cell here that you see has both a red receptor and a green receptor. Understand? Good. Okay, so let's say that the sympathetic division gets activated. This is going to cause release of a neurotransmitter called norepinephrine. Norepinephrine binds to receptors called adrenergic receptors, and what you see here is that every cell in the SA node possesses these adrenergic receptors. That is going to make these cells targets of norepinephrine and targets of the sympathetic division. So what's going to happen is that when these neurotransmitters bind to these receptors on the SA node, that's going to produce the response, which is that the heart rate starts to go up, like what you get with exercise or with stress. Okay, but what if we activate the parasympathetic division instead? The parasympathetic division is going to release a different neurotransmitter, in this case acetylcholine, which binds to a class of receptors called cholinergic receptors. The particular one we're looking at here is called a muscarinic receptor. So acetylcholine here is going to bind to its receptors, and you'll notice the response now is going to be exactly the opposite. The heart rate goes down, very similar to what you see when you are resting and relaxing. So the idea here is that these cells of the SA node have receptors for both acetylcholine and norepinephrine, but we tend to only release one neurotransmitter at a time, so the very fact that these cells are targets for both divisions allows for them to respond to signals coming from both divisions. So this concept of dual innervation of organs and tissues like the heart that are simultaneously controlled by both sides of the autonomic nervous system, this is something we will talk about much more in the future. For now, I just want you focusing on the concept of ligands binding to receptors and eliciting responses. So just some takeaways before we wrap up here. It's really going to be critical for you as you continue your study of human physiology to always be aware of which cells in the body have receptors for a particular signal. If you're focusing on insulin signaling, you need to be aware of which cells are going to have insulin receptors. If you're focusing on adrenaline or epinephrine, you ought to be aware of which tissues in the body have epinephrine receptors in order to have a complete holistic appreciation of what that hormone or what that neurotransmitter is going to do throughout the body. So one last thing I'll leave you with here, let's talk about a hormone called glucagon. You will learn all about glucagon in the future, but for now, the idea is that glucagon is secreted by the alpha cells of the pancreas when blood glucose starts to get very low. What glucagon will do is go all throughout the body through the bloodstream, bind to target cells that have glucagon receptors, and the response is going to be that blood glucose starts to shoot up to get back into the normal range. But you'll notice this graph here, which is showing you the relative mRNA expression for the glucagon receptor gene, is not equal between all these different tissues. The tissues that have a lot of glucagon receptor mRNA is the liver, adipose tissue, and the kidneys. All the other tissues that you see here have little to no glucagon receptor mRNA expression, meaning that if they don't express the mRNA, they're not going to make the protein. So basically what this is going to tell you is that tissues like the brain, the GI tract, the heart, the pancreas, and the lungs, these are not targets for glucagon, whereas the ones with the green bars are targets for glucagon. Now, I've adapted this graph from information that you can find on the NCBI website, which stands for the National Center for Biotechnology Information. So if you're ever curious about which cells in the human body have a particular kind of receptor, NCBI is a great resource to go to. Okay, that's going to do it for this video. Here is some vocabulary I think you ought to be aware of. And for checking your understanding, number one, what determines whether a cell can respond to a given hormone or neurotransmitter? Number two, can activation of a receptor produce more than one kind of response? And number three, how does the combination of ligand and receptor help to determine the nature of the response? Okay, that's going to do it for this video. If you have any questions, go ahead and drop them in the comments section, and I will see you